Okay, so as Dave mentioned, I'm at UMass Amherst in the Department of Environmental Conservation in the Massachusetts Cooperative Fish and Wildlife Research Unit. And I just finished my PhD in December. And a lot of my work was focused on the Brook Floater, which is a freshwater mussel. And the focus of my work was really on population restoration. So that's when you grow mussels in the lab for the purpose of putting them back out into the river. And so my dissertation really focused on laying the groundwork for that to try and understand what it would look like to do that in Massachusetts. But often when I presented my PhD, I found that I was already a step ahead of everyone else because I was jumping to this population restoration component. And most people were thinking, well, why are we restoring mussels? Why are we putting all this effort into restoring mussels when we don't know why we're losing them, right? So some species are rare now, we're losing populations, there's lower numbers of individuals. And so this talk is a little bit about why that might be. And so this isn't something I focused on for my dissertation, but we've drawn a lot from the past 6 years of working with Brook Floaters to start to discuss some of these things. So the Brook Floater is a freshwater mussel in the order Unionida. There's approximately 900 species worldwide. They're on, I'm just gonna get rid of my own video so I don't have to look at myself. They're on all continents except Antarctica, and they can have these really fun morphologies and different colorations, and these can actually differ based on the river system, which makes their identification kind of challenging. But what makes these mussels especially interesting is their life cycle, which I'll talk about in a moment. So in North America, there's approximately 300 species. It's the most biodiverse continent. And as you can see, this is on par with amphibians and reptiles and even mammals. But freshwater mussels also have a really high rate of impairment. So there's approximately 30 species that have gone extinct just in the last century. So we're really living through this extinction in this taxa. Here in Massachusetts, there's 12 species of mussels, and half of them are unlisted, so they're not on the Massachusetts Endangered Species Act. They include the eastern pearl shell, eastern floater, eastern lamp mussel, the triangle floater, eastern elliptio, and the alewife floater. And then there's 3 species that are of special concern, and that's the creeper, the tidewater mucket, and the eastern pond mussel. And in Massachusetts, there's 3 species that are endangered. So that's the yellow lamp mussel, the brook floater, and then the dwarf wedge mussel, which is the smallest mussel and is also federally endangered. So even though we don't have a really high diversity of mussels here, we can find them in a lot of different habitats. So some species are habitat generalists. We find them in the Miller's River, and they're also found in ponds. There's also the eastern pearl shell that we can find in really small streams. They do better in environments with less nutrients. Just want to try and move this down. It's okay, so mussels are also ecologically important. They filter water up to 8 gallons a day. And but this depends on the species, depends on the temperature and the flow rate of water. But large mussel beds are also really important in rivers because these can actually stabilize the substrate. So if we look at this mussel here, it's anchored into the substrate, so its foot is, we can't see it's in the sand. And then what we see is the siphon, so the in-current siphon here where it's drawing water in, and then the ex-current siphon. And so dense mussel buds really help to stabilize the substrate and serve as habitat for other macroinvertebrates. They also can support microbes that actually remove nitrogen from the rivers. So freshwater mussels have this really unique life cycle, which I hinted at earlier. And how that works is they're separate sexes. So the male will release sperm, and then the female will filter the sperm through, which fertilizes her eggs. And these eggs develop into larvae, which are called Glaucidia. And they look like tiny Pac-Men. They're just about the size of a grain of rice. And the female mussel has a job to try and attract a host fish, because these are actually parasites. They need a fish in order to develop. And so once the mussel attracts the fish, it will clamp onto the gills and it will obtain nutrients. And it will stay on the fish for a couple weeks to a month, at which point it will drop off as a small juvenile. And this juvenile isn't yet filter feeding, so it's feeding with its foot, which is called pedal feeding, and then it develops and becomes a smaller version of the adult. So I'm going to show you a video of a mussel specialist trying to attract a specific kind of fish. I know, that's weird. That's not. Hopefully it will disappear with the mouse off of it. Okay, sorry about that, everyone online. So this, for anyone who missed the video, I can send the link after too. But it shows a mussel attracting a fish in order to put its Glaucidia on the fish so that it can transform into a juvenile. And so this image is showing multiple lures. So the letters depicted here are showing different lures that the mussel is using to try to attract a host fish. Really just trying to get that host fish to attack the lure, and then it will release its Glaucidia. And then in examples E and F, these are mussel generalists. So it means that they can use many different kinds of fish species to transform their Glaucidia into juveniles. And so in example E, this mussel is broadcasting its Glaucidia into the water, hoping that a fish will just swim through it and the Glaucidia will attach. And then in example F, the mussel is releasing this Glaucidia on mucus strands. And then the fish will swim through this and become entangled, and they'll attach to the fish. So everyone here will know this really well. So for my work, I focused on assemblages, mussel assemblages in streams. And one in particular, which consists of the eastern pearl shell, the triangle floater, keeper, eastern elliptio, and brook floater. So these species are found together in many areas, or there's subsets of them in different rivers. And of this assemblage, brook floater is the only one that's endangered, so it's declining at a rate that's higher than the other species that it co-occurs with. So the brook floater is found in the US from south in Georgia all the way north to Maine. and then in Canada and New Brunswick and Nova Scotia. And throughout its range, it's critically imperiled, imperiled, or not ranked because there's not enough information on the species. In Massachusetts, there used to be more populations than there are currently. So the red are populations that have been extirpated. And in the green are the 4 remaining populations. So West Branch of the Farmington River, Bachelor Brook, the Ware River, and then the Nissitissit River. And the top causes of population losses or declines in the number of individuals is recruitment failure from a host fish. So that just means that the mussels could still exist in the stream, but they don't have a host to transform their Glaucidia into juveniles. It could be from water pollution. So here's just a screenshot from when I was on an airplane. I took a picture out the window. But this is just an example of, the intense development that we have in some watersheds and all the diffuse stressors or pollutants that can enter rivers. And then destruction of habitat types. So in-stream changes to the channel can change the flow and the sediment characteristics. And this change in habitat can affect what mussels can live there. And then an overall restriction of ranges. So if a species can't inhabit or disperse into new habitat that could be available, so if they don't have a host fish, and if there isn't good habitat, then there's a strict restriction in their range. And now we're also facing climate change. And so for species that are at the edge of their ranges already, then we're seeing that there could be a reduction in the number of populations. And then also that can lead to a lack of genetic exchange. So if there's multiple mussel populations in a river, they can, and they're connected through host fish, then there can be a transfer of genetics. And, but without that connectivity, then there's no genetic exchange, and that can really weaken the population's ability to adapt to changes. So this brings us to our question, why is Brook Floater rare? Is it because there's a lack of host fish? Does water quality negatively affect their growth and survival? Does available physical habitat limit their distribution? So is there just not enough good habitat in Massachusetts for them to live in? And then could stream barriers restrict their genetic exchange? So are there populations, but they're just not interacting with each other? So we'll just go through these questions, relating it to what we've learned about Brook Floater in the past 6 or 7 years. So Brook Floater uses a technique called passive entanglement. So they release those mucus strands that have Glaucidia in them. And this is a picture of a Glaucidia, and they actually have these hooks on them, which help them to really latch onto a fish and embed in it. But it doesn't hurt the fish, really. The fish is fine and continues to live. And we found from previous work that Brook Floater can use a lot of different fish species. So these are all laboratory experiments where people go out and they collect fish and they bring it into the lab and they collect the mussels. And they found that Brook Floater can use many different fish species to transform those Glaucidia into juveniles. And here in Massachusetts, we've done 3 different experiments looking at what hosts might be compatible with Brook Floater. And so I went out and I collected adults. So they have this bright cantaloupe colored foot, which can be actually really vivid depending on the river. And we didn't have enough of the broodstock in Massachusetts, so I actually had to go to Maine to collect some of these. And then I extracted their Glaucidia, and I wanted to do this without harming the adult because we placed all these adults back out into the stream because they are so rare. And then we basically just make a Glaucidia bath. So we mix the Glaucidia in water in a bucket, and then we add the fish. And then we put these in tanks pictured here. So all of this work was done at a U.S. Fish and Wildlife Service facility called Cronin Aquatic Resource Center, which is in Sunderland, Massachusetts. And in these tanks where the fish are, at the outflow, there's these mesh bags, and that's where we capture the Glaucidia, which then metamorphose into juveniles. So here's a picture of undeveloped Glaucidia, and the fish will start shedding these for the first few days. So we'll collect a lot of just undeveloped Glaucidia. And then as they stay on the fish, they'll metamorphose, and then we start to get these small juveniles. And this is their little foot here that you can see. They're actually very adorable. So we found in Massachusetts that this species was compatible with all the fish we tested, so all 10. And so this is just a graph showing the proportion of metamorphosis on that fish. So how many of those Glaucidia metamorphose into juveniles? And here's an example of 3 of the fish species in each of the 3 experiments. So in some of these experiments, we retested the same fish species, but there's a lot of variability. So long-nosed dace has a lot of variability even between replicates. So this is between each tank. And then Siamese sculpin is not so much. It's pretty consistent. We collect a high number of juveniles between experiments and replicates. But then brook trout has this really high proportion of metamorphosis, and then we retest it and it's really low. So even though it's using these fish, the fish seems to differ. Even, this is the same species, so there's a lot of variability that we don't understand there. What does this mean ecologically? These experiments are done in a lab, and we tested some fish that are cold water specialists, for example, sculpin, and their habitat just would not overlap with habitat where you find brook floater in the wild. So just because we did these experiments doesn't mean that the mussel is using them as host fish in the wild. And there has to be overlap in where they exist in the water column. So if the fish is always in the upper part of the water column, it probably wouldn't interact with the mussel, which is more benthic in the sediment. So here's a picture of another species, Crassus, which is one of the most endangered mussels in Europe. And this study tested 2 different populations of this mussel within the same river. So there's 2 populations, they're in the same river, but they were isolated, so they're genetically distinct. And then they tested the same fish species with the same populations, and they found different results. So even though these mussels can use many different fish species, it seems like they may still be co-evolving with what fish are most present in the streams. And so when we're trying to do laboratory experiments, it's really difficult to understand where that variability is coming from. So for our first question, is there lack of host fish? Maybe, we don't know. There could be, because it's really hard to understand these things that are happening in the wild when we're doing experiments in a lab. So our second question is, does water quality negatively affect growth and survival? So most of my work was spent growing mussels. It's very time consuming, and they're very needy. They're just always dying. So most of my time was spent growing these mussels in the lab. And then a small amount of my time was spent doing experiments with them. So this is one of them, in which I tested the water quality and how it related to the mussel's growth and their survival. So these systems are called silos, and they're made of concrete. They're basically a salad bowl size. And through the center, we put this chamber. And how it functions is there's upwelling that goes underneath the silo. So there's an opening right here. And then the mussels that are in this chamber interact with the water. They get oxygen and nutrients that way. So in those 4 rivers where we find Brook Floater, we put out 4 of these mussel silos. And in each silo, we put 5 age 2 mussels and then 8 mussels that were younger. So they're 1 year old. And we put these out for 1 summer. So from June to October in 2019. And then we visited these locations every other week, and we collected a number of different water quality parameters. We collected information on food. So we used chlorophyll A as a metric of algae, which mussels filter and use to grow. We measured the mussels, so the shell size determined their change in growth. We deployed temperature loggers, and these recorded temperature every 15 minutes. And just from prior knowledge that about these 4 rivers, I was going into the study thinking that Bachelor Brook, the mussels we put in Bachelor Brook probably wouldn't do very well because in that river, just from previous reports, we've already lost some populations there within the river. And there's only 1 population remaining. That river also can get really warm, so the temperature there is significantly higher than the other 3 rivers. Whereas the Nissitissit River and the Farmington River, the watersheds are less developed, and I thought the mussels would have higher growth and survival in those rivers. We did see high survival for all rivers, which is good, over 90%. But remember, I'm limiting these mussels to only interacting with water quality. So it's good that they're able to survive for this short period of time, just in the water, not in the sediment. And here's an example of a mussel's growth over the 16-week period. So each mussel is individually tagged in this with glitter because they're so small. So this glitter happens to be black, but you can really see the change in shell growth with these bands right here. And here's the total growth. So this is the total amount of growth for these age 1 mussels across each of the 4 rivers. So Bachelor Brook, the river that I thought was going to do the worst, had the highest growth. And then the Farmington and the Nissitissit had significantly lower growth, which is puzzling, right? Because not what we were expecting. But so if we look at what's going on in Bachelor Brook, and we look at how the growth rate relates to some of the parameters that we collected, we can see a positive relationship. So here is the growth rate of the mussel. So this is how much the mussel is growing each day and its relationship to food. So this river happens to have more algae. So more food, higher growth, that makes sense, right? And then it also has this interesting relationship with temperature. So there's a positive relationship with temperature, which usually holds true for species. When there's warmer temperatures, there's greater growth. But it seems to reach almost a threshold here, after which higher temperature doesn't do the mussels any good. There's actually lower growth. So this river does have high temperatures. That could be a factor affecting populations. But it's so puzzling, right? So then it begs the question of, is greater growth good? So is that a good proxy to judge a river? And here's an example of this, of a species from Alabama. I promise this is the last graph I'll show. This is a species from Alabama, and it has 2 populations. And one of these populations, its length, its growth is really fast, but then it dies much sooner. So there's a trade-off. It's growing really fast, and then it has a shorter lifespan. But in this other population of the same species, it has slower growth, and then it lives much longer. And so it seems like brook floater are just adapting to whatever stream that we put them in. And so greater growth in bachelor brook might not be good because it could cause them to have a shorter lifespan. And overall, that could cause, population declines. So for our second question, does water quality negatively affect growth and survival? Maybe, it might. We did collect a lot of other measures and found that there were some other relationships that might be causing lower growth, and that's due from other like de-icers entering the river. So potassium, calcium, things like that. So our next question is, does available physical habitat limit distribution? So habitat for mussels is really hard to understand because it's nested across multiple spatial scales. So from a landscape scale, a river is affected by everything that's going on in the landscape, right? So potential runoff or other biological attributes, so dispersal of certain species into the watershed, that's affecting the river. And then it's also within the river, the habitat, there could be a really nice patch of habitat, but mussels can't get there because there's limited dispersal. So there's no fish for them to be able to travel to that new habitat. And then habitat is limited by the local environment. So once the juvenile falls off the fish, can the mussel survive there? Is there everything that the mussel needs to live in that little sandy patch? So I looked at the abundance of brook floater across mesohabitats. And mesohabitats are sections within a river. So here's the main channel of the river. And then there'll be areas of riffles, runs, or pools. And a riffle is just a shallower section of the river. It's usually, there's a cross section. They're usually rocks protruding, and we don't usually find as many mussels in those areas. And then a run is a relatively even flow. It can be deeper. And then a pool, of course, is deeper than a riffle and a run and usually has slower flow. And so I looked to see if maybe mussels had higher abundances in any of these different mesohabitat types. So we went out and we snorkeled, we measured out 100-meter reaches, and we assessed the mesohabitats for vegetation, for large wood, for other habitat that we thought might influence the mussels. And we sampled across the state in these 4 larger watersheds, and we essentially sampled 145 mesohabitats. And we found that there was no difference in mussel abundance among mesohabitats. So even though we were in all these different rivers, there really wasn't a significant difference. So it seems like they were using many places in the river. But we did find that substrate was important at predicting mussel abundance. So in areas where there was really heterogeneous substrate, so there was sand where the mussels could burrow in, but then larger rocks that might be stabilizing the stream bed, that's where we found the most mussels. So for this third question, I looked at mussels at within a stream reach, and we found that it's difficult to understand their habitat. But all the stream reaches that we did sample were relatively lower moderate gradient rivers, which is where it generally has lower flow. And so for general physical habitat, we can kind of identify those rivers, but then within those rivers, it's really hard to find mussel beds. So this brings us to our last question: could stream barriers restrict genetic exchange? So this is an interesting one for Brook Floater because we don't have any data on this at all. We've never investigated this. There's a larger effort right now being done by the Fish and Wildlife Service that's collecting genetics across Brook Floater's range. But as far as differences in populations within a river, you know, we're not sure. But Mass Wildlife has been doing a lot of work with dam removal. So some of you might have heard of the Millie Turner Dam removal. This happened in 2015 and was in the Nissitissit River, so that most northern population. And there was two populations of Brook Floater, and they were separated by the Millie Turner Dam. And so this was a big effort by DER and Mass Wildlife, and they relocated those mussels that were downstream of the dam and brought them upstream where the other population was, and they've actually been tracking that population long term. So they're still working on that data, but it's I think the restoration process it's a project that you have to track for a long time to really see recruitment or differences. And so dam removals like this, where it connects populations, are, you know, really helpful, especially for increasing genetic exchange. But one thing that I found while I was doing a lot of my habitat work was that I kept finding some of the best or the densest Brook Floater populations around dams, which is counterintuitive of what you would think, right? So it's kind of unexpected habitat. But they were primarily around these run-of-river small mill dams, which of course we have a lot of in New England. And they, how they function in a river is more like the natural flow. So it will just, there will be the sediment buildup here, and then kind of a lower dam height, and it will be natural flow over this. And some papers have found that mussels like this habitat because mussels, so we find Brook Floater sometimes right below these dams. And that's because in these small impoundments, there can be higher temperatures, greater food availability that are transferred to the mussels downstream. And then also with climate change, sometimes these pools are in the impoundment, if we find mussels here, are areas that still have water during droughts. And so these are usually just smaller dams that we're finding them around. And it just goes that, you know, dam removal, especially when there's imperiled mussels around that are using that habitat, just brings in a whole other component for management or decision maker trying to make decisions on what's best for the watershed, which is why I'm not in management, I have to make those decisions. So for this last one, it's something that's interesting to me, but yeah, that could also be a factor because we don't have information on those populations and how they may be distinct. So this brings me to stressors. What's causing all these declines? It's probably different based on location of the Brook Floater population. So just broadly categorizing a stressor that's causing all the mussels to decline probably isn't the case, right? These are probably really unique situations to where the populations are. And when there's multiple stressors, it's hard to identify which ones are causing lower population numbers. So because of all of those unknowns and the effort that it takes to try and understand why mussels are declining, I focused my dissertation on population restoration. And so that's growing mussels and then trying to find habitat where you think there's no more threats and then exploring putting mussels out there. So it's really laying the groundwork of trying to understand population restoration for Brook Floater in Massachusetts. So here's some other great mussel resources. So the book is up here, so please take one. It's by Ethan Nadeau in partnership with the Connecticut River Conservancy. There's also the Freshwater Mollusk Conservation Society website. Mass Wildlife has some really good fact sheets for each of the species in the state. And then the state biologist created this iNaturalist for Massachusetts. So if anybody uses iNaturalist, there's a Massachusetts Freshwater Mollusk Survey Project, and you can enter your observations there and he'll check your identifications. So my research was done with a lot of collaborators, and here I just listed 5 who are especially important. So Allison Roy is my advisor at UMass Amherst. Peter Hazelton used to be the Mass Wildlife biologist, but he's at the University of Georgia now. He's heavily involved in a lot of mussel work in Mass. David Perkins runs the mussel lab in Sunderland, where we grow mussels. And then Andy Fisk at the Connecticut River Conservancy was really involved in the habitat work. So the whole team of CRC was instrumental in helping me work with volunteers for a lot of that work. And then Tim Warren at the Fish and Wildlife Service is a biologist there, and he has been really helpful with everything. So a lot of funding partners, USGS, UMass Amherst, a grant from the Massachusetts Environmental Trust. So people who buy those license plates, the Fish and Wildlife Service, and Mass Wildlife. So I will end with this especially unique mussel behavior of using a host fish. So this mussel is called an Epioblasma, and they stay in streams. They're just kind of open like this, and then their lure is in the center, and they try to draw the fish in to attack the center of the lure, and then on contact, the mussel will snap shut on the fish, and it has these teeth or denticles that clamps onto the fish, and it holds it, and then it pumps it with its glaucidia. So it looks like this fish might have been trying to get away, like it was turning around or something, but there's all different types of methods that mussels have used to try to attract fish. This is another one. With that, I'm happy to take any questions.
Can you play that video that was again?
Yeah. Yep.
I'm not sure whether we determined whether the people at home got to see it or not.
Yeah, I think we should play it here if we can.
Yeah. So everyone on Zoom, we're just going to play that video again in case anyone missed it.
In the streams of Missouri lives the Lampsilus mussel, a simple animal with an extraordinary life cycle. To reach adulthood, its young must spend part of their lives inside a fish, the largemouth bass. To get there, the mussels must make physical contact, a difficult task, as mussels don't swim. But the bass has a weakness. It's a voracious predator of small fish, particularly darters. Even the slightest wriggle of a darter's tail will attract bass. Believe it or not, the fish on the mussel is an imitation, a perfect replica that will lure bass within striking range. The mussel can somehow sense approaching fish and wriggles its lure faster to entice them. If it gets the twitching just right, the remarkable lightness should do the rest. On impact, the mussel squirts its young into the bass's mouth. These snap shut on the gills like spring-loaded traps. Here they stay, drawing blood from the fish until several weeks later they drop off as tiny, fully formed mussels. Also a favorite prey of the bass are these striped shiners, and some mussels mimic them. Considering mussels are blind and have never seen a shiner, the likeness is incredible. The eyes, fins, and even the stripe look just right, yet the mussel knows nothing of its own appearance. These lures have evolved because bass more often attack mussels that look like fish, so fishy-looking mussels need more descendants. After millions of years of blind evolution, this process of selection has turned mussel flesh into a lifelike lure. It takes a good imitation to fool a bass in clear water, and some of them are incredible. This starter mimic even has a mouth which goes. This mussel is the same species, but its curious leopard print design may not find a taker, and its genes will go no further. This lure looks pretty good, but the bass is unconvinced and turns it down. Mussel lures are constantly improving, but fish are getting ever better at recognizing face. It's another arms race, and it's still creating diversity in the streams of Missouri to this day.
Thank you.
Yeah, it's a neat video.
Okay, tell them to ask questions in the chat, so we can answer. For you can.
I'm like, why am I typing? I'm happy to take questions over Zoom if